Hello friends, and welcome to my video lecture series. Our topic for today is human embryonic development up to Blastula stage. Okay, so let's start with the topic. uh, First of all, the zygote, which is formed due to fertilization of the sperm and the ovum, is basically a single cell. Okay, so what happens is the sperm fertilizes the ovum and a single cell is formed. Okay, so this is the outermost covering of the ovum called as the zona pellucida. So all of you are well aware of that structure, and inside it is a single cell which is present. Okay, this is the zygote. All right, so now what happens is this single celled zygote undergoes many divisions which are also called as cleavage. So what is a cleavage? Cleavage is nothing but a mitotic division that is responsible for the single celled zygote to get converted into a structure which is having many cells. Okay, so you can say that the zygote, which is a one cell structure, gets converted into a two cell structure, which gets converted into a four cell structure. Then from 4 it gets converted into a 8 cell structure. From that it gets converted into a morula. So morula is a 16 cell structure, and from there on, over a period of time, it will get converted into blastula, which is having around 100 cells. Okay, so single cell, from that to around 100 cells, and the blastula then changes and undergoes modifications to form a gastrula. So these are the embryonic changes which take place in a single celled zygote. These mitotic divisions are called as cleavages, and the resulting cells which are formed, this is a single cell zygote. Here there are two zygotes, so each of those miniature zygotes is called as a blastomere. Say for example, this is single zygote. After cleavage it forms two zygotes. These zygotes are called as blastomeres. So here there will be two blastomeres, four blastomeres, 8, 16, around 100 blastomeres. Okay, each of this blastomere has the capacity to form a completely new individual. Each blastomere is behaving like a miniature zygote. Now we need to remember, till this blastula stage, this zona pellucida remains intact. So this structure is going to uh, remain the same in shape and size, and inside this structure the resulting blastomeres which will be formed will get smaller and smaller because the space is going to decrease. Okay, so new cells form in order for them to accommodate inside the space of the zona pellucida. The blastomeres will keep on getting smaller. I hope you understand that. Okay, so if you compare the number of blastomeres in a morula, that is 16, with the blastomeres in this two cell stage, here the two blastomeres will be larger in size, whereas here the 16 blastomeres will be smaller in size. I hope you understand. I hope you are understanding what I'm trying to say. Alright, so each of this cleavage gives rise to double the number of cells. These are called as blastomeres. The zona pellucida remains intact, so the entire structure of the embryo will remain intact up till blastula. In blastula, the zona pellucida disappears. Okay, so let us just draw a schematic diagram. So what happens is, after about say 30 hours of fertilization, this is the zona pellucida. What happens is the single zygote undergoes cleavage to form two large blastomeres. So this is called as the two cell stage, that is after 30 hours of fertilization. Alright, then what happens is, after say 60 hours of fertilization, that two cell stage becomes a four cell stage. So again the zona pellucida remains constant. Now there will be four blastomeres, so these four blastomeres will be smaller compared to the last two blastomeres. Each one will have a nucleus, so this is called as the four cell stage. Then around after 72 hours of fertilization, that is after 3 days, again the zona pellucida will remain intact, and you will have 8 blastomeres. Alright, so this is called as the 8 cell stage. So every time in every cleavage, basically mitosis is occurring, the cell number is doubling, and the genetic material is being distributed equally among all the blastomeres. Alright, these individual cells are the blastomeres. I hope you are getting the point. Alright, this is the zona pellucida, alright, which is remaining intact. Now these cells will keep on dividing, and the next division which will occur will give rise to a cell which is a 16 cell stage. Again the zona pellucida is intact, so I'm going to draw 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 cells. So this 16 cell stage is called as a morula. Do you get me? Alright, so that is basically a morula, and what happens is this morula will then convert, or the cells of the morula will get differentiated. So first of all I'm going to draw the zona pellucida as usual, and after that the blastomeres get differentiated into two types of cells. One is called as the outer layer of cells. So I've drawn 10 cells over here, alright, and I'm going to draw 6 cells inside. So this inner layer of cells is also called as inner cell mass, and this outer layer of cells is called as outer layer. This is the zona pellucida. So this is the early morula. I'll write down early over here, and this is the late morula. So the late morula has 16 blastomeres. It is a 16 cell stage. It has the zona pellucida. The cells or the blastomeres get divided into two regions, the outer layer of cells and the inner cell mass. Okay, now what happens is this morula reaches the uterus in about 4 to 5 days. So what's happening is, when these cells or these blastomeres are being formed, the zygote or the embryo is traveling through the fallopian tube, and from the fallopian tube it is passing to the uterus. So it takes around 4 to 5 days for the morula to reach the uterus. And what happens is, as soon as the morula reaches the uterus, this outermost zona pellucida which is present, it disappears, and these outer layer of cells absorb the fluid secreted by the uterus. Okay, so there is the inner lining of the uterus which is called as endometrium. So endometrium basically secretes endometrial fluid, and this fluid is absorbed by this outer layer of cells and they become flat. So there is a space formed between this outer layer and this inner layer of cells. Okay, so what is going to happen is, I am going to still take that yellow color. Okay, and the zona pellucida has now disappeared, so I'm going to draw this outer layer of cells. Now this outer layer of cells which are present are called as trophoblast cells. Do you get me? And they keep on absorbing the liquid secreted by the endometrium or the inner wall of the uterus, and what happens is this inner cell mass moves to one end of the trophoblast cells, and there is a cavity which is created over here. So that cavity is called as the blastocele. So cele comes from coelom, which is the true body cavity. So this space is blastocele. These are the inner mass of cells. Okay, but now this inner mass of cells or inner cell mass is called as embryonal knob. I hope you are getting my point. So what happened was, when the morula reached the uterus, the endometrium, the inner wall of the uterus, this zona pellucida disappeared. These cells, outer layer of cells, absorbed fluid from the uterus and they became flat, and because of that a space was created inside which was called as blastocele, and this embryonal knob are the cells which are the inner cell mass which have moved to one end. So now what happens is, this structure which is formed is called as a blastocyst or a blastula. So the next stage after morula is called as the blastula. This blastula will increase in size since it doesn't have to restrain itself inside the zona pellucida. So that structure is called as the blastula. This structure, inner mass cells, is now called as embryonal knob. These outer cells, which were earlier outer layer of cells, are called as trophoblasts. These are sticky cells. So these trophoblasts help in attachment to the inner layer of the uterus or the endometrium. Okay, so zona pellucida was preventing the attachment of these cells at an improper site. Now its work has been done, so it disappears. So this stage is called as the blastula. The cells of the trophoblast surrounding the inner cell mass, these cells are called as robber cells, or these are also called as cells of robber. Robber is the name of the scientist. Alright, so we have the robber cells, the trophoblast cells, and the inner cell mass and the blastocele. This stage is called as the blastula stage. Okay, so summarize whatever we have learned. This is the diagram. So initially we began with the zygote, which was a single cell stage. After 30 hours the first cleavage or the division took place. It gave rise to a two cell stage in which two blastomeres were formed. After 60 hours of fertilization, 4 cell stage. After 72 hours of fertilization, the 8 cell stage, which gave rise to the 16 cell stage. All the blastomeres were present together. The 16 cell stage is called as morula. This morula again develops to form two layer of cells called as the outer layer of cells and the inner mass of cells. What happens is, this morula reaches the uterus in about 4 to 5 days. The endometrium secretes a fluid which is absorbed by these outer layer of cells, and the zona pellucida disappears. So what happens is, there is no zona pellucida. Because of these outer cells absorbing the fluid, they become sticky, and these are called as the trophoblast cells. The space is created called as blastocele, and now this entire structure is called as a blastula or a blastocyst, which increase in size. These inner mass cells are called embryonal knob. These are the cells called as cells of robber. So I'll highlight the cells of robber to you. These are the trophoblast cells surrounding the embryonic knob, so they are the robber cells. This is the blastocele. Okay, so friends, I hope you have understood. You can always pause the video or rewind to read back the information. Okay, so thanks a lot, and hope the concepts regarding the development up till the blastula stage have been cleared. Okay, so thank you for listening to this lecture patiently. Anybody, if you need extra information, alright, so here is my email address. You can always contact me over here. You can ask me any queries, or if you need to know any more details. So it's biren.daftari@gmail.com. Okay, thank you.